So welcome back. We are now in part 2 of the Cryopreservation for Regenerative Medicine workshop. My name is John Draper and I'm the Program Director of Research and Partnerships at the Stem Cell Network. For your information, part 1 is actually now available on SCN's YouTube channel, along with lots of other great content. And at some point in the near future, we'll also post most of the content from part 2 today. So in a moment, I'll post the workshop booklet link in the chat, and we will you'll be able to share that and access it. A word about the session. Please consider this to be an interactive session. Use the Q&A function to ask questions of our speakers as they talk. Those questions will be gathered by the chair and addressed to the speakers at the end of their talk. So please do use that. If you possible, please use the Q&A function and not the chat. Sometimes the chat is used, and we prefer to use the Q&A because they can actually answer written replies as well. Breaks are scheduled, and I'll let you know about that later on. And as you're aware, this is being recorded, so I've already said that this will be available on our YouTube channel. So with that, I will hand over to Dr. Alexandra Stolzing, who is she holds a PhD from Humboldt University, Berlin. She recently worked at the Cryopreservation Research Institute in the UK and as a group leader and deputy head of department at the Fraunhofer Institute for Cell Therapy and Immunology in Germany. She was tenured in 2018 as a full professor of biogerontological engineering at Loughborough University in the UK. And in addition, she has worked as a VP for research and a CSO in startup companies in the rejuvenation biology sector. Alexandra, thank you, and please take it away.
Yeah, thank you very much for the nice introduction. So today we will have one session on the cryopreservation of complex systems. Really an area of research that needs progress and is riddled by problems. Since like the dawn of organ transplantations in humans, which started around about 1906, when the first renal transplants into human was performed, people have been dreaming and also testing to actually fully cryopreserve organs for the transplantation sector. But organ transplantation is not the only complex system we are interested in. The regenerative medicine field has been progressing very, very rapidly, and more tissue engineered complex systems are coming alive and have been used in humans. And today we will have a couple of such examples. We have Dr. Sascha Reichmann from the Institut of Vision from Paris, France, that will tell us a little bit about his work in retinal organoids. He has really pioneered some work on human-induced pluripotent stem cell differentiation into these organoids, and using them as a test system really requires nice cryopreservation of these systems to have them readily available. So let me hand over to Dr. Sascha Reichmann. He will be doing the first presentation, and hopefully he's live and ready to roll.
Yeah, thanks. Hello, everybody. First of all, I want to thank Dr. Glyn Stacy for its invitation to this workshop, and I want to thank also Dr. John Draper for the organization. I present you today experimental result of cryopreservation. You have seen theoretical explanation of cryopreservation, and to illustrate this, I will talk about simple retinal organoid cryopreservation. I will begin with an introduction of retina, followed by the generation and culture of retinal organoid from human induced pluripotent stem cell, Ipscs, their cryopreservation in detail, and the analysis of their phenotype after freezing thawing, and I will finish by a little conclusion. The retina is the innermost light-sensitive layer of tissue of the eyes. It transfers light ray to electrical information interpretable by the brain. and some age-related of inherited retinal diseases allow photoreceptor loss, the light-sensitive cells, and therapy to avoid cell death depend on the state of the disease. For example, neuroprotection still could maintain present photoreceptor, while in the severe case, prosthesis, gene, or cell therapy are needed. To find neuroprotective molecule by drugs,
Sascha, you're not sharing your slides. We're not seeing anything. Sorry for interrupting.
Oh, sorry. I'm. Sorry. You see?
Yes, thank you.
I'm sorry. Bon. Generation and culture of retinal organoid. I'm cryopreservation, freezing and thawing, and Eros phenotype analysis. The retina is the innermost light-sensitive layer of the eyes. It transfers light ray to the electrical information interpretable by the brain. Here, some age-related and inherited retinal diseases allow photoreceptor loss, the light-sensitive cells, and the therapy to avoid cell death depends on the state of the disease. For example, neuroprotection still could maintain present photoreceptors, while in severe cases, prosthesis, gene therapy, or cell therapy are needed. To find a neuroprotective molecule by drug high-throughput screening, for example, or to have sufficient cells substrate for cell therapy, we need retinal cells and better human retinal cells. To produce these cells, we have developed a new retinal differentiation method of Ipscs based on our simple method by passing embryonic body formation and using only media changing on adherent Ipscs. With this protocol, after 1 month, emergent retinal organoid can be picked in dishes and cultured in floating condition in plates. During this 3D culture, retinal progenitors, named Ipscs, composing structures can differentiate into all retinal cell types of human mature retina, either 4 types of photoreceptors, but also in neuron integrating the photoreceptor signals and forming the optical nerve. Cell remplacement represent a potential vision restoration strategy, and prior study in animal model revealed that transplantation of post-mitotic photoreceptor precursor isolated from mouse neonatal retina may be feasible. Using our retinal differentiation protocol, we showed that photoreceptor into this structure express the cell surface antigen CD73 previously described for the enrichment of mouse stem cell derived photoreceptor. We demonstrated that CD73 based magnetic associated cell sorting can select photoreceptors, and the transplantation of this photoreceptor precursor in a rat model of retinal degeneration supported the possibility to develop a cell-based therapy. Considering the potential use of photoreceptor precursor for future cell transplantation and the length of the differentiation, we thought to develop different cryopreservation approaches to store organoid along the differentiation processes. The first strategy was to cryopreserve whole retinal organoid at stage of development where CD73 is well expressed, close to day 100 in culture. And the second approach was we examined whether a dissociated retinal cell from organoid could be cryopreserved. For this lecture, I want to describe you the different steps allowing retinal organoid cryopreservation. Thus, organoid was removed from the culture plate and put directly in the cryotube at room temperature. And after a few seconds, organoid sediment at the bottom of the tube, and culture media is replaced by cold cryopreservation medium as a cryostore from stem cell, initially developed for stem cells, but which work very well with another cell types or tissue as organoid. And about a cryostore, cryostore is composed by DMSO at 10% and another ingredient, but I'm sorry, I don't have more description of medium composition, but we can at minima think that trehalose were present in the formulation because this complex sugar preventing ice formation, thereby water is replaced by trehalose, is often used in stem cell vitrification. Cryopreservative media must to be used at 4 degrees. At this step, it's very important to avoid absolutely temperature increase. After the tube were placed in the freezing container at -80, allowing a slow temperature drops by 1 degree Celsius by minutes. And be careful, stock your freezing container at 4 degrees before use. Quite often I see in culture labs containers in bench. So if you place your favorite cell in the counter at room temperature, you will have surprise and bad surprise when you will thaw cells. After minimum 4 hours to overnight, place as soon as possible your tube in the -150 or liquid nitrogen. Don't forget the cell at -80 because at this temperature the cryopreservation time is reduced, and after some weeks the cell will be damaged. To conclude fastly this step, your handling time here must not exceed few minutes. Therefore, it's important properly prepare the cryopreservation protocol. For thawing, at this step, it's not the moment to discuss of the last series in Netflix because normally after 6 minutes and 30 seconds, you can do it. Take out your tube from the -150 and place it in water bath at 37 degrees during more or less 30 seconds. And until to have a little ice, maintaining the cryopreservative medium at low temperature around 0 degrees Celsius. After, dilute the cryopreservative medium with culture media, wait aero sedimentation, remove the all supernatant, and resuspend the organoids in a pre-warmed media, and put the organoid directly in your culture plate with an equilibrated and pre-warmed medium. Alors, the result of this cryopreservation. Here, the retinal organoid was cultured in suspension until day 84, where where the photoreceptor is present and was examined by using immunostaining 16 days after thawing at day 100. In control case, fresh organoid photoreceptor precursor express CRX, photoreceptor precursor marker, rocoverine, and but also the CD73. Frozen retinal organoids showed also the presence of CRX and rocoverine positive cells. And of course, interestingly, the CD73 positive cell, considering as the transportable cell population, were still present. Quantification of CRX cells at day 100 did not reveal any significant difference between the number of cells in control and in frozen organoid. In the second approach, we tested whether dissociated retinal cells from organoid could be cryopreserved. In this case, retinal organoid until day 84 or day 100 were dissociated using papain, and retinal cells were cryopreserved using the cryostem freezing medium. After thawing, cells were plated in specific coating cover slip and cultured for 5 additional days in vitro. Immunostaining relieved that frozen photoreceptors have kept their CD73, rocoverine, and CRX expression as non-frozen dissociated cells in both cases, in both time. Here too, the quantification showed no significant difference between in number of double positive CRX and rocoverine cells between frozen and control cells. Here I want to present you a new thing. It's a long-term culture of cryopreserved organoid. This organoid was cryopreserved at day 96 and thawed for long culture in floating condition. You see that after 5 days after thawing, the marker CRX and rocoverine is not well expressed, but after 1 month and a half, we can see a recovery of this expression. Nevertheless, after 1 year in culture, very long, culture, some apoptosis take place and few cells express the specific marker. You have here the phenotype of the organoid after 500 days, more than 500 days, and here at 100 days, 15.4. And for mature marker like opsins, we observe maturation of photoreceptor cells which don't express the opsin at day 100 as known in fresh non-frozen organoid, but which express this specific protein, this specific opsin after 1 month in culture, showing the possibility to do the maturation after culture, after thawing. Sorry. Alors, to conclude, retinal organoid can be cryopreserved, and the initial phenotype can be preserved. Organoid can mature after thawing, And the cryopreservation is compatible with long culture approach, but you see some cell death in very, very long culture. About protocol generalities for organoid cryopreservation, respect slow freezing and fast thawing, and you can use anti-apoptotic molecule to overpass cell stress at the thawing state. And for me, it's very important to master the cryopreservation to have success in cell therapy. It's why I have words here. Mastery the cryopreservation will direct the cell therapy success, of course. If you have a lot of cells, but you can't preserve them, it will be difficult to make something. I want to thank the Institut, the old director José Alassal and the new director Serge Picot, the S2 team and my group, and also the stem cell network for this invitation. Thank you.
Thank you, Sascha. That was a wonderful presentation. And we currently have only, now 2 questions. So the first question was if you were using differentiated iPS cells, and let me expand that question to have you work with other donors, like have you worked with animal iPS cells, and are they behaving differently during cryopreservation?
If I have tested on organoid from animals other than human, is this? No, I produce only human organoid, and I'm sorry, but I think it's.
That's no problem. We have a second question here, and that is, what is the purpose of having some eyes left in the tube after the thawing? There was this nice little picture that you had with an ice cube in it.
Sorry, in the.
What is the purpose of having some eyes left during thawing?
The ice in the tube.
Yes.
Yes, it's very important to keep the ice in the tube. I can share this. Sorry, where is my presentation? You see my presentation or not? Yes.
Not yet.
Not yet.
Now.
Sure. Not. Sorry. PowerPoint. Yeah. At this step.
Yes.
We need to keep the little ice in the tube to maintain the cryopreservative medium at 0 degree more or less, because you damage your cell or your tissue if you use a cryopreservative medium at room temperature or of warm. Thus here you can dilute your cryopreservative medium with room temperature media to dilute the cryopreservative medium and to avoid action of cells. And fastly you remove the supernatant and you change the medium by a fresh medium at room temperature. You increase a little bit the temperature, and after you put directly at 37 degrees to recover the structure. I think it's a point very important when you thaw the cells of organoid or other cells or structures. To maintain the cryopreservative medium cold.
Okay, so we have another question, and that is asking towards the therapy treatment. Are organoids preferred over dissociated cells?
Today we test dissociated cells dissociated cell because it's better for injection in subretina. If you want to use directly organoid, place tissue from organoid in subretinal space, it's more difficult to develop devices to place correctly the structure in the subretinal space. It's why for this first development, we used the cell suspension, but existing paper describing organoid transplantation. And if I remember well, the organoid creates some rosette in subretinal space. It's not the better thing, but it can work after development.
Okay, we have another question here that is asking about the cell density of organoids, and maybe also like what is the organoid concentration in your solution? Like what is the largest size of organoids that work?
You can, I put between no more 10 organoids in 250 microliter of medium. And in one organoid, you have million cells, 1 million cells, depending the time, but in the organoid at day 100, after 100 days in culture, the organoid contain 2 million, 3 million cells.
It's a great slide because the next question is about like, so which one is the preferred cryopreservation solution that you would advise for people to use? Is there one that you find more like working better than others?
I have used the Cryopreservation meta that I use for Ipscs. I have not tested another one because this works very well. For me, a CryoStor or medium dedicated for Ips or stem cells, it's very nice because it's neutral and to avoid differentiation of this normally pluripotent stem cell. It's why I have used this. You have, but when you cryopreserve organoid, you can see some cell death inducing by cryopreservation, but not a lot with this cryopreservative medium. I have used this because it worked very well on other cell types like retinal pigmented epithelium or another cell types. I like but this is the Canadian product.
Okay, so let me finish with one last question that was asked, and it is, what is your take-home message for the audience?
Take home.
What is the most important point?
The most important, I think, it's the rapidity, it's the rapidity of the cryopreservation. You need to manipulate fastly when you want to cryopreserve cells and organoid. Here you see it's only 2 or 3 minutes in the bench to cryopreserve your cell. You put fastly in the freezing container, and in the same idea, when you thaw your cells and your organoid, you need to work quickly. Here it's 30 seconds, and after 2-3 minutes, and another step of 2-3 minutes. Here in less than 5 minutes, it's done. For me, it's the principal point.
Well, then these are the concluding words. Thank you very much for your presentation. And then we will be moving over to the next presentation. So if you could stop sharing your slides. And we have next Dr. Jason Acker. He will be talking about the donor factors and cell heterogeneity in cell cryopreservation. He is a professor at the University of Alberta, and has been working in the field for many years, and hopefully will have some great insights into like what are the important factors. He has written over more than 200 publications in the field and is very active in his consulting and advising companies also on the establishing on biobank and cell therapy programs. So with that, Dr. Jason Acker, if you're ready, yes, I see you're ready. Then let's start.
Well, thanks, Alexandra, and thanks everyone for joining us today with this important workshop on cryopreservation. I'd just like to thank the Stem Cell Network and the Society for Low Temperature Biology for putting this all together. So today I'm going to sort of merge the talk on donor factors and cell heterogeneity as it relates to some of the complications that we have when we're developing cell therapy cryopreservation strategies. But I will also weave in on request some of the slides that I was unfortunately not able to present at the last session. So you will get to see a snapshot of some of that work as well. So I don't need to really introduce to this group the requirements and then the desire to have optimized cryopreservation strategies for the cell therapy and regenerative medicine industry, but just want to reinforce that the cells and tissues that we're cryopreserving will be in various states depending on their pre-processing, where they're sourced from, what stage they are in the engineering process, and then ultimately what their target application is. So, there's no one solution that's going to fix all of the challenges here, but the need to be able to preserve these for an extended period of time is what's common, and cryopreservation is often the technology that's looked to achieve that. So I'm going to start with a discussion around cord blood, and then I'm going to transition into a discussion about some other cell therapies. So with umbilical cord blood, HPCs, there are a variety of different methodologies that have been adopted over the years, right from the Hal Broxmeyer's method and the Rubenstein method to others over the last few years. And in a lot of cases, if you look at the literature, you'll see data that looks something like this. Now, there'll be a generic description of the cryopreservation method that's used, which typically includes, 10% dimethyl sulfoxide and perhaps some other additive like a plasma or a dextran, and a generic cryopreservation dump, freeze, and storage of liquid nitrogen and a thaw, and then some sort of cryoprotectant removal process. And even though the methodologies sound very similar, the recovery that's often reported is variable. Everything from 100% recovery, everything's perfect, to something that's less perfect in terms of the recovery. Now, I'm not going to go in and discuss about some of the challenges and opportunities that still exist in terms of standardizing the methodologies that are used. There has been an enormous amount of work in the industry to look at single platform methodologies for measuring cell recovery and viability, particularly around membrane integrity. But what I want to highlight here is that the outcomes can be variable, and that shouldn't be surprising to us. In the previous workshop, we discussed some of the basics of cryobiology. And one of those was the fact that each cell is going to have a different response to a standard cryopreservation process. So I want to thank John Boast, who put together this data a few years back, where he used a standard cryopreservation protocol, a 10% DMSO, 1 degree per minute. And he looked at a variety of different cells that are cryopreserved in that solution. And then he also looked at using a hypothermazole type solution or a cryosterol solution. And in this table, he quite nicely shows the viability differences that can exist across different cell lines. And that can range from 18% to 90%. So even though you're using a standard protocol, you're getting variable responses. And there's a variety of explanations for that. And I'm going to go in and talk a little bit about the role that cell heterogeneity and donor factors has in influencing this. So we know from the previous workshop that the response of cells is going to be dependent on a variety of factors, the cooling rate, the cryoprotectant concentration that's used, and each cell is going to have an optimal recovery depending on how you treat those cells. And cells like ova or oocytes can have an optimal cooling rate that's in the sub 1 degree per minute, whereas cells like red cells, their optimal cooling rate in the absence of cryoprotectants is in the order of hundreds of degrees per minute in order to get an optimal outcome. Now, of course, adding cryoprotectants will modify these kinds of curves and flatten them out to give us a different kind of response. But in the absence of cryoprotectant, we quite easily see that different cells are going to have a different response. And that's not surprising because the cells are very different. So if I just compare and contrast a red cell with a oocyte, the red cell morphology is clearly different. Its volume is different. Its surface area is very different than a oocyte. And the cryopreservation protocols that have been developed to optimize these two different cell types, which in a lot of cases have been two of the most successfully cryopreserved cell therapies. Red cells, over 130 million transfusions every year. It is the single largest cell therapy that's used on the planet. Cryopreservation is achieved using a glycerol solution, slow cooling to -80, and rapid thawing in a 37-degree water bath with multi-step removal in order to remove that glycerol. Whereas oocytes, where there's been well in excess of 2,000 babies now born from cryopreserved oocytes, human children, the cryopreservation protocol is very different. Ethylene glycol is the optimal cryoprotectant with sucrose. Rapid cooling is used in the order of thousands of degrees per minute. Storage of liquid nitrogen and rapid warming as well in order to actually achieve successful cryopreservation of those oocytes. Now, the cryobiology behind all of this was introduced in the previous section, but what I'd like to really start to just emphasize is in addition to the structural changes that these cells have, there's also very specific molecular differences, and the membrane is one of those that is really critical, and the structure of the membrane is obviously related to the function of that cell. In the case of the red cell on the left-hand side, you have a structure that really is designed for those cells to move throughout the body and deliver oxygen, and as well as have other immunological functions in the body. So deformability and permeability of those cells as they transverse organs with very different osmolalities within the interstitial or within the intravascular space is really critical. Whereas in the case of the oocyte, you have a membrane structure that really is around protecting that oocyte during its development and preparing it for fertilization and then division and growth. So it's a multi-layered protection system that is enriched in the energy sources that are going to need to sustain that embryo, the developing embryo post-fertilization. Now, one of the things that was discussed in the first seminar was really the role that the cell's osmotic response plays in the design of an optimal cryopreservation solution. So just to quickly review, cells will shrink and swell in response to that changing environment that the cells find them in during the cryopreservation process. Whether that's the addition of the cryoprotectant, the cells will shrink and swell if that cryoprotectant is permeable, or swell if it's not, or shrink if it's not permeable. And then subsequently during the freezing process, as ice forms in a pure state and water is removed from solution concentrating the solutes, the cells that are exposed to those concentrating solutes are going to respond osmotically. And those processes are reversed during the thawing and warming process. So here's just an example of a cell that is subjected to a hypotonic or a hypertonic solution, and the changes that occur that we would normally like to see in the time scales in which that occurs is relatively quick, but still significant. And these processes are temperature dependent. So one of the questions that was in the previous talk is why do we only thaw the cells to 4 degrees or just the presence of ice removal? Well, if you thaw to 37 degrees, then the process of removing that cryoprotectant solution is going to be very different. And in many cells, the case, a single step dilution of that cryoprotectant at a high sub-zero temperature will result in the cells lysing because of the change of temperature on the permeability characteristics of the cell. So, this is fairly well described in the cell systems and has been used effectively to design cryopreservation protocols that are optimal for cells with very different permeability characteristics, as has been discussed. So I'm going to talk a little bit about red cells and some of the work that we've done to really look at now how heterogeneity and donor factors plays a role. So the red cells are very unique in the sense that they are extremely fast responders due to the presence of transporters on the surface of those membranes. So in order to actually measure the permeability characteristics, we had to develop a stopped flow system that is able to measure the quenching of hemoglobin in the millisecond time scale. And this was developed by one of my PhD students and published in 2014. There's other methodologies that are used to measure the permeability of cells. Coulter counters are often used or as a methodology or other methods. But in the case of red cells, we had to develop a unique method because of the speed at which they respond. Now, when we had this method, one of the things that we wanted to, that Maria, Dr. Jarova was interested in was the development of a cryopreservation process for cord red cells. So these are the red cells that are found in the cord blood that are often discarded during the production of the hematopoietic progenitor cell product. But there is an interest in the cryopreservation of these cords as a potential therapeutic. So her work was to compare the permeability characteristics of adult red cells. So these are adult red cells that are mature, that contain hemoglobin A, versus the cord red cells that are of a different development stage than the adult and have hemoglobin F typically present. Now, these are cells that, morphologically, you'd be hard-pressed to be able to differentiate them on a microscope, but physically have very different properties. And as we highlight here, the permeability characteristics of the cord blood red cells are very different than the permeability characteristics of the adult red cell. Yet, these are all red cells. They're just of a different development stage. So, this was very enlightening because it tells us that, if we are going to develop a cryopreservation process for cords, we can't simply mirror what we would do with an adult red cell because those cells, even though they're red cells, are very different. Now, the other thing that we observed when we were actually doing this work was donor dependent. So when Dr. Jarova was looking at her data, she was collecting red cells from different donors. And on various days, she would measure the permeability characteristics. And she started to see differences across the donors that she was actually measuring. So these were 7-day-old red cells that were exposed to a hypertonic salt solution at 24 degrees, and the permeability characteristics were measured. So, when we looked at the run-to-run variability, so if you take the same sample from the same donor and run it, there's a natural error in the ability to determine the permeability characteristics due to an issue that I'll talk about in just a minute. But if you looked at different donors, you see a much larger variability, which really raised the question, these are red cells that, from a transfusion perspective, we would treat as if they were the same product in terms of how they're used. But depending on the donor, they have very different permeability characteristics, which, as I've alluded to, cryopreservation and the optimal development of a cryopreservation strategy requires you to understand the permeability characteristics. What role does this donor-to-donor variability have on the optimal response that you're seeing using a standard methodology, cryopreservation method? We went on to further look at other factors like the donor age and sex and the method in which the red cells were manufactured. And lo and behold, we found differences as well. So not only does there is variability across donors, that donor variability may very much be influenced by the age and sex or gender of that donor. So this work has really been informed by some more recent work that Dr. Mikhailova has done recently, looking at the subpopulations of red cells that naturally exist in a blood product and naturally exist in people. While we like to think that red cells are all the same, red cells from the time of their development and maturation coming out of the bone marrow through to their senescence and final removal from circulation by the reticuloendothelial system, they're changing. They're not the same cells that they were over that 120-day period that they'll circulate in our body. And in fact, there's a number of factors that we've identified, including donor age and sex, that is influencing that. So what in a single sample, you're going to have young cells that are a few hours old, and you're going to have red cells that are 100 days old or 120 days old. So how does that influence then the characteristics of that product? So Dr. Mikhailova has been really focusing on trying to pull apart how these cell populations are really influencing the aggregate properties of a cell. Now, bringing this back to cryopreservation, some of the recent work that we published just last January, this was collaborative work with Dr. Higgins and Benson, looking at seeing if we can develop a more rapid method for deglycerolizing red cells that based on mathematical optimization of the process. And Dr. Higgins' group did a phenomenal job really developing some methodologies, but when we went to actually test those methodologies with real samples, what we found was that there was a significant contribution of donor variability to the optimal cryopreservation response, or the ability to deglycerolyze those using an optimized response, which really highlighted the fact that that if we are going to use a standard deglycerolization process, that the variability, the natural biological variability that's going to exist in that sample is going to be important. And the study further raised the, I guess, the suggestion that, in fact, within a single red cell sample, there are subpopulations of slower responding red cells, meaning red cells with different osmotic permeability characteristics, which hamper our ability to really use the most rapid methodology for deglycerolizing. And in fact, we have to use a much slower process in order to account for the slow response of those subpopulations. So, and this is within red cells, which for many of us, we would think red cell is a red cell. And that's clearly not the case. So, what are the implications for this? I think, one of the things that we have to remember is that when we're developing a cryopreservation process, there are multiple steps that are involved in the development of this process. Everything from the choice of cryoprotectants, the rate of removal, the temperature of nucleation, the cooling rates, the storage temperatures, the warming rates, and then subsequently the cryoprotectant removal. All of that is dictated by the physical properties of those cells. The permeability characteristics, their susceptibility to the toxicity of the cryoprotectants, and the presence and formation of ice that it can occur inside and outside of those cells. Now, when we developed this protocol, we developed for a cell type. But what the data and what our work is really showing is that in addition to cell type, we need to be considering the variability or the heterogeneity that exists within that. And then if we're talking about human-derived or animal-derived cells, where it may be difficult to standardize the production, the downstream or the upstream production of those cells, to standardize their physical characteristics, then we're going to have to deal with the natural subpopulations that exist in those samples. So just to add to the list of variables that become important, and this list builds on the presentations of the previous session. When we have temperature, there's a variety of factors that we have to consider when we're developing an optimal process. I would just like to add, at the bottom, the other biological factors, which now are becoming just as important. And those are not only the biochemical and physical state of the cell and the hematocrit, but now we're really having to need to consider heterogeneity as we're developing our optimal cryopreservation processes. Now I'm going to switch gears a little bit and just finish this up by talking about this a stability study that we actually have been looking at and the importance of stability. So obviously when we store our cell therapy products, we're storing them for a purpose. And a lot of times that purpose is time-based. We want them to be stable over a period of time so that we can actually separate from in time and space the donors or the manufacturing of those cells and the recipient. One of the things that's often forgotten in the application of cryopreservation to cell therapies is that there is a challenge of freezing, and that challenge is really the lethality of an intermediate zone of temperature, typically between -15 and -60, that the cells must transfer where significant attention needs to be paid if we are going to minimize the damage to those cells. During the natural storage of cells, there is transient warming events that can occur. This occurs during natural events in your biobank operation, whether it's compressor cycling, filling events, inventory management. All of us have been in the lab and seen students pull open the liquid nitrogen, pull up the cassette and look for that box and start to look for that cell and get it in. And that process is introducing transient warming. It is warming those samples. Now, when we were interested in this, we went to our biobank and our cord blood bank and really introduced this concept. And when we talked to the technical staff, this is the kind of response that I heard, and I often hear from companies and organizations I work with, is that transient warming doesn't occur in our samples. The products stay frozen for the duration of their storage until we thaw them. Well, no, we went and actually looked at that, and this data, I'll just go back, I will go through this data quickly, but if you are interested, it is summarized in a recent cytotherapy paper that came out. In this work, we basically went and measured how fast samples actually warm in a cord blood unit. And I think the technical staff were quite surprised that the samples, when they're taken out, will warm up to a potentially damaging temperature within 30 to 60 seconds of being removed. So, that's a relatively fast time. So transient warming does occur, and that is a reality of many biobank operations. Now, the question, the next issue is really, when we presented this data, again, the technical staff said, well, our procedures state that we should move quickly. So it's unlikely that these samples are out long enough. So again, we went back and did a time study where we actually measured how fast staff are actually moving samples and removing segments. So one of the processes is within a cord blood bank is to remove a segment for compatibility testing or quality testing. Well, when we measured the time it took across sites, it was taking 1.5 minutes or longer to get that sample out of liquid nitrogen, take the segment, get it back into liquid nitrogen. Now, this time does not include the cooling time back to a stable storage temperature. So in fact, this would be much longer than the time we're reporting here, the actual time that the cord blood unit is actually cycling. So staff aren't as fast as they think they are. And these processes, this transient warming during their manipulation. So, the next question is really the functional question. So the samples warm up. That shouldn't really affect the product much, should it? And that was really a rhetorical question. So we went again and actually looked at that. And I won't go into the detail here, but we looked at 3 different scenarios that replicated the movement of a cord blood unit within the bioprocessing lab, and was really based on real times that we measured during our time studies. We processed those units, we split them, and then we did standard quality assessment. We were interested in measuring the quality in different parts of that component. So the 20 mil main component within the cord blood unit, the 5 mil, and then the segments as well. And I'll just highlight that this work was done, was really led by Dr. Nick Pinot at Canadian Blood Services. So when we looked at the data, not surprising, the total nucleated cell recovery was very good. It doesn't seem to really be significantly affected. But when you start to dive into the data and start to look at the viability and recovery of the CD45 cells, you start to see differences in those 5 mil and segments of the cord blood unit. When you look at the CD34, again, CD34 cells are very robust and very tolerant of cryopreservation stress. So we didn't see a lot of differences in terms of membrane integrity, which is what viability is measuring here. But we started to see some interesting data on the recovery. And most interestingly, when we actually take those cells and look at whether these cells are actually potent and can do what they're supposed to do, here's where we saw a significant difference in both CFU GMs and CFU totals, which tells us that transient warming of these samples is having a detrimental effect on the potency of the cells. So even though you don't pick it up in your standard recovery and counting methodologies, when you actually look at the cell potency, you start to see a significant difference. So the study conclusion was that really transient warming of products occurs quickly during routine operations, that transient warming can negatively impact the quality, recovery, potency, and viability of the cells, and that biobanks really need to look at taking steps to mitigate exposure to these transient warming events if they truly want to have stable products for an extended duration. So I'll end there, and I'd be happy to answer any questions.
Thank you very much. That was a very comprehensive presentation of all of the problems and challenges in banking. We have some very quick questions that we can answer now. So there was one question of the red blood cell cryopreservation. Addition to the 40% glycerol, is there also 10% DMSO in the cryopreservation?
No. So red cells do not respond favorably to DMSO. In fact, DMSO solubilizes the membranes of red cells. So we do not use DMSO for red cell cryopreservation. It won't work. We use glycerol, and there's both a 40% method and a 20% method that, again, rely on changes to the cooling and warming rates in order to achieve the optimal outcomes. Traditionally, most blood services around the world are using the 40% glycerol method.
Okay. There was one question around the difference that you have been observed in the membrane permeability in the cord in adult red blood cells. So what does it actually mean for the patient? Is one better than the other? Like what kind of like functional difference is there?
Yeah, good question. The permeability characteristics really come into the design of the cryopreservation strategy. So while there absolutely are going to be impact on permeability, but more in case deformability of those red cells in terms of their ability to transfuse and circulate within the body. In the case of the permeability characteristics, we're really more concerned about how quickly glycerol can get across the membranes and how quickly water moves across those membranes, so that we can optimize that glycerolization and deglycerolization processes. And the fact that the cord have a much faster permeability really means that there is a significant risk of damaging the cells if you add the cryoprotectant too quickly, or if you attempt to remove the cryoprotectant too quickly as well. That can have some detrimental effects on the quality. So we had to optimize our addition and removal strategies based on this data.
And then maybe a last question, and it is, does the storage time actually change the permeability of the cord red blood cells?
Yeah, so I assume you're speaking about the storage of the blood in the blood bag yes, over the 42 days versus the frozen storage. So yes, and for a whole bunch of reasons. So during the hypothermic storage of red cells for 35 or 42 days, depending on where you are in the world, those red cells undergo a hypothermic storage lesion where they do actually change. And one of the changes that occurs is to the membranes and to the morphology, deformability, and permeability of those cells. So as a result of that, the protocol that you use for cryopreservation may depend on how long you've stored those cells. And I won't have time to go into the detail here, but one of the things that we know is, for example, if you're manufacturing a CAR T cell, the expansion processes that you're using for your CAR Ts is designed to optimize the recovery of cells. So you may be developing a protocol that produces a recovery of cells and targeting a phenotype. Finding is that perhaps the longer you put those cells in culture or when you're doing your stimulation, that could significantly affect the permeability characteristics of those cells. And as a result, their ability to respond to the cryopreservation process that you're actually using. So it's not simple. It's not a simple idea or a simple goal of trying to maximize the recovery and of your phenotype cells. You may be adversely affecting their ability to respond favorably to a cryopreservation process. So some of the challenges that organizations have as they're scaling up their manufacturing, as they're going from pilot scale manufacturing, small volumes of 1 mil to scaling up their manufacturing to produce a 50 mil or 100 mil product, is that they neglect to consider the impact that extended manufacturing process is having on the physical characteristics of the cells. That's going to affect their ability to respond to the cryopreservation protocol that work really well when you're freezing 1 mil cryovials, but may not work very well for the larger samples. And there's been some very classic failures of scale up when you're designing your cryopreservation processes because organizations fail to consider this.
So what is a standard volume of blood then? If something like this actually exists, is there a standard volume?
So what standard volume of a blood?
Like a blood bag?
There is. So yeah, there's international standards around this. Typically, a red cell product will be anywhere from 250 to 200 to 300 milliliters. That's being cryopreserved for a red cell product. There are groups that freeze smaller volumes for certain applications, but a typical volume of a product is in that 250 50 to 300 milliliters. And the cell number, again, the cell concentration is going to be dependent on the red cell concentration. So the hematocrit is about 55%, and the volumes, like I said, about 300 mils.
Awesome. Thank you very much for this enlightening presentation. And we will be moving on then to the last presentation in this session. And this will be Dr. Juan Medrano from the University of Nottingham. He's a veterinarian that works in cattle fertility, and he will be talking about the cryopreservation of the uterus.
Okay, well, thank you very much to Jonathan and Alexandra for the introduction and for the presentation, for the invitation to present this. So I'm not a specialist in cryopreservation, but we've been working for a few, well, say for a few years, really, not many, on cryopreservation of different reproductive organs. So we have done ovarian cryopreservation. We usually do ovarian cryopreservation, tissue cryopreservation, and the whole ovary. And in this case, I'm just going to be presenting some of our attempts to do a full organ, another reproductive organ cryopreservation, which is the uteri. And in this case, we are using the sheep as the model. So this is kind of the idea of the things that I'm going to talk about. And what I'm going to present to you today is going to be a series of there were a series of studies that we did along probably a couple of years. And we're hoping to just to present the final idea. So this is kind of the initial steps to looking into uterine cryopreservation. There are many things to fix, of course, as Alexandra mentioned earlier, and I think we hopefully will start working on those at some point. Okay, why the uterus? Well, we know the ovary. We used to work specifically on ovary, and we understand the importance of the ovary. But why the uterus? Well, the uterus is, without the uterus, you cannot carry out a pregnancy. So the uterus is the organ that holds the fetus and holds the fetal development. So that means from implantation, from the start of pregnancy all the way up to the end of pregnancy and parturition. So this is a very complex tissue, and the uterus usually, there are two main parts that are important, which is the glandular portion, which is the endometrium, which allows the implantation, produces the nutrients to keep the embryo, regardless of the species, at the start of the pregnancy. And then, well, once placenta is formed, then something has happened. But endometrium is very important. That's the glandular portion of the uterus. And then the myometrium. And the myometrium is the one that contracts, allows the contraction, and especially is important for at the start before parturition, at the time of mating, and also is very important at parturition, because that's the one that allows the fetus to be expelled. And then we have a perineum, which is just, a perimetrium, which is just the serosa that surrounds the uterus. So we have several tissues within the same. There are some cases, and this is probably about 3 to 5% prevalence in humans, where patients are either present without a uterus or with a partial uterus, and those usually are caused of uterine infertility. So the most critical of this is the absolute uterine infertility or factor infertility, the IUFI. So that's when the patient doesn't have a uterus, doesn't have a full uterus, or only has partially developed that uterus, and that can be a congenital problem. There are several syndromes in humans that cause that. Or in some cases, there has been an acquired, or the uterus has been removed because of other issues. So it might be because of cancer, the uterus needs to be removed. Or sometimes at parturition, there are some bleeding, profuse bleeding during the parturition, that the only way to stop that and to save the mother is to remove the uterus together with the fetus or with the newborn. So those extreme cases where the uterus is removed, then there is no longer a uterus. And the only way that you can treat for these cases is to do a uterine transplantation, because there is not really any other way to have a pregnancy if you don't have a uterus. And the only other option that patients have in this case is either go for surrogacy, so borrow a uterus from somebody else, or adopt, which is the two options. But if you want to have kind of carry out your own pregnancy, this is a bit difficult. So Uterine transplantation, I think, has been developed for now is currently developed. I think in there the group in Sweden from Dr. Brandstorm have been developed this one, and I think in 2014 was the first one, the first successful transplant, followed by a pregnancy and parturition with those patients. Usually what happens is that the uterus is transplanted and then is removed at the time of parturition, but I think, as far as I know, that's the way that is been doing on uterine transplantation. There are two options for these patients. Either you go for a live donor, so in that case you will need to have simultaneous surgeries happening in the same building sometimes, so they can have one group of surgeons removing the uterus, and it needs to be really careful because you need to be careful around the blood vessels, and then another group of surgeons preparing the receptive, the person that will be receiving the uterus, the receptor. So in this case, this happens at the same time. So usually they are very long surgeries. The other way is that you can collect the tissue and move it to a different place. And for those cases can be also a live donor or can be a deceased donor, a recently deceased donor. And I think the Brazilians have done this, a group in Brazil, and they have shown that the pregnancy coming from a deceased uterine donor in 2017. When this happens, the uterus needs to be transported on cold ischemic conditions, and it can only be kept for a very short period of time. Usually it's 1 to 2 hours is the common one, but it can be kept out to a day. But of course, the longer you keep it, the more damage or the more ischemic problems you can get into these tissues. So just to keep in the mood of this workshop, is cryopreservation a possible viable alternative? We believe it might be, because that will give us the chance to have uterus as a banking uterus. So we have the organs ready for transplantation when it is necessary. That would be one option. The other advantage might be that you can properly run a pathogen screening, and probably most important, the donor recipient match. So you ensure that there is less instances of rejection, and of course reduce immunosuppression if that's possible, because you will need to keep these immunosuppressive drugs throughout pregnancy, and sometimes the immunosuppressive drugs that are used are not very compatible with reproduction. So sometimes that can be that needs to be taken into account. And then going back to what the problems will be, is that you will be needing to cryopreserve several different tissues, and probably the most important ones is preserving the glandular function, the muscular function, and also the vascular part of the uterus. So there are several tissues that you need to cryopreserve, and as mentioned by Professor Acker, every single tissue, every tissue has different requirements for cryopreservation. So you need to kind of manage to find this sweet spot between everything to allow the recovery of the function. Some of this work has already been started in sheep. So in sheep in animal models, sheep and pigs, they have been done some experiments in this species, usually using fast perfusion, 50 ml per minute, and the common 10% DMSO, which I think is similar to the one that we use. So after following cryopreservation of these tissues, there seems to be a recovery of the contractility if you stimulate it by oxytocin of the uterine tissue, with the myometrium. There are the tissue seems to be viable. There are some metabolic viability indicators, like the uterus is still uterus, so the uterine cells are still metabolizing glucose and are still producing energy. So it seems that they are alive. So that's quite good. Some of them have tested survival by using fluorescent staining, and again, you have a very good survivability or survival after cryopreservation, but very few have tested what happens with the vascular part once you do the cryopreservation. Okay, so that the overall study, the overall objective of our study was to try to evaluate this in the sheep as model. So we tried to evaluate the actual damage that is happening in the tissue and the functional damage that is happening to the tissue following cryopreservation. So we use a slow freezing cryopreservation system, which is similar to the one that we use in whole ovarian cryopreservation. So these are the early stages of this. And we think that the sheep is a very good model in the case for human to mimic the human uterus in terms of size. But we understand that the sheep has a very different reproductive processes. So they don't really have a cyclical endometrial shedding. So that might be one of the issues, and that's why recovery of endometrium is quite important in human, and also the myometrium is quite important in case of sheep. That's the case. So for this, as mentioned, we use you can see my screen. So we use the similar protocols to those that we have reported in the ovarian cryopreservation, the successful ovarian cryopreservation, and we collected sheep uterus from the abattoir. So we collected the uterus. Immediately after collection, they are transported to the lab. Of course, we did not collect the 115 at once, so it's been a very long, over 2 or 3 years that we've been working on this, so it's a cumulative number, so don't worry, it doesn't take that long. Once in the lab, the both arteries, the both uterine arteries are cannulated directly and perfused with heparinized ringer for about 10 minutes, and the perfusion is at 0.5 mils per minute. Following that, we start the cryopreservation perfusion. So we do a cryoprotectant perfusion through the vascular system. And this is quite important for uterus, as you will see later on. So we perfuse them at different times. And following this, we either some of those after perfusion, some of those tissues, as the numbers are in there, cannot see them very well with the screen. So it's about 6 per group, where the tissues were equilibrated with deuterium and used for neutral magnetic resonance to evaluate the DMSO pickup of the tissue. And then the remaining tissues were cryopreserved and then thawed, a rapid thawing methodology that we use. And then the tissue was evaluated for the cytotoxicity, was evaluated using lactate dehydrogenase release, blood vessel integrity using microspheres, and then fluorescent microspheres, and then the functional activity using endometrial production of prostaglandin F2 alpha and myometrial viability, so the myometrial contractility. And of course, we use several controls for that. So what we found was, as I suspected, and something that we have already observed in the ovaries as well, is that there is a linear relationship between the uptake of cryoprotectant, in this case DMSO, and the time that you keep it in perfusion. So in this case, as you see, the blood vessels and the myometrium seem to pick up most of it in a linear manner, depending on the time of perfusion, while although there is an increase in the endometrium, there is not the same amount of DMSO that was present in the endometrium. And that makes sense, because what we did was perfuse through the arteries, through the uterine arteries, and not through the cervix. And that's why the central portion of or the last tissue that will be perfused will be endometrium, which is towards the center of the uterus. So that's why we believe that might be the case. The other thing that we observe, and this is quite interesting, is of course cryopreservation caused a lot of damage. So the tissues that were cryopreserved had a higher cytotoxicity levels of LDH, so a higher release of lactate dehydrogenase, which was kind of expected. But the interesting bit is that there seems to be a linear, like a negative linear relationship between the LDH production and the time in the cryoprotectant. So the longer you keep them, the less cytotoxicity or the less LDH release that you have from the cells. However, this only applies kind of to the myometrium and endometrium, which are tissues that are after the perfusion tissue, which is the vascular tissue. That's why the vascular tissue seems to have a lower damage, let's say. we know that we are perfusing through the vascular system. So when we start perfusing vascular system, if you don't keep an eye on the pressure that you are perfusing, then you end up causing some vascular problems because of the pressure. So what we wanted to do was evaluate if we were causing that, so the endothelial expansion, if you want, and then trying to see if we were causing a large damage into the blood vessels. So we use fluorescent microspheres. In this case, we use 3 sizes, the green one, which is a 10 micron microsphere, the red ones, which are a 1 micron microsphere, and then the blue ones, which are 0.1 micron microsphere. So we were thinking that if the larger, the 10 microns or the 1 micron were seen in the tissue, so in this case, we're seeing the lumen is around here, and then you see the wall, the vascular, the blood vessel wall in there. So you see the lumen there, and then the wall, the blood vessel. So what you see is the large microspheres remain within the lumen, so there is no really much of that damage of that size. But the small ones, the blue ones, they tend to form this kind of dual line or dual side line in the endothelial portion of the vessel. So there seems to be there is some extravasation of the smallest fluorescent microspheres. In this case, we see that that seems to be the case, especially in tissues that were not cryopreserved, that they were not really infused with cryopreservative, with DMSO. But the longer you keep them in there, the 40 and 60 minutes, there seems to be a decrease in that extravasation. However, when we did this same analysis, but just following the first 10-minute perfusion with heparin isolant, we already started to see the dual lines on the either side of the blood vessel. So we think this might be some kind of mechanical effect of perfusion. So we might need to adjust the perfusion rate in this case, because that seemed to be the cause rather than the cryopreservation itself. In terms of the functional activity of the tissue, what we did was culture the fragments of the tissue, small portions of the endometrial tissue, in this case, endometrium, just to evaluate the production of prostaglandin F12 and see if the endometrium is affected. So what we observe, of course, is that cryopreservation reduces the viability of the endometrium, but it doesn't reduce it that much as we expected. So we thought that it would be completely decreasing the production of PGF2 alpha, but there is a recovery. So that seems that there is still about between 70 to 80% of the tissue is still responding and producing prostaglandin F2 alpha. So that gives us a good indication that although the endometrium is the one that seems to be receiving less of the cryoprotectant, the endometrium seems to be quite robust and keeping it functional. Then we wanted to check what happened with the myometrium in those tissues. So we already know what happened with the vascular vessels. We already knew what happened. Well, we were already finding out what was happening with the endometrium, the actual endometrial activity, and then we just wanted to see what happens with the contractility of the tissue, with the contractility of the myometrium. So we initially did a bolus oxytocin dose. So the bolus oxytocin, it should increase, or it should induce a peak contractility in the myometrium. And that's exactly what happened in the controls. As you see, there is an increase in the peak, in the contractility or in the contraction of those of the control tissues, which seem to be continuous up until 10 minutes. We stop at 10 minutes, but the tissue seems to be still contracting after that. However, in terms of the cryopreserved tissues, what we observe is that they contract initially, but then they seem to plateau, and they just keep at that plateau and then they relax. So that happens with the 40 and 60 minutes, but if we keep the tissues longer than that, then it's exactly the opposite. So it just never contract. So it has a very small peak, but then immediately after that it drops. So there seems to be an effect as well of length into the functionality of the muscle. Similar, we did some experiments on ex vivo in an organ bath, also using the oxytocin directly into the tissue, into strips of endometrial and myometrial, the really myometrial myometrial strips. And what we observe in those organ bodies is exactly the same that we expected, which is the control reacted immediately after oxytocin. But although there was a contraction following the oxytocin in the cryopreserved tissue, the reaction is a lot smaller, or is not as large as it should be. So it's still active, it's still responding, but it seems to be a bit damaged. And this was even visible before the actual oxytocin bolus. So we follow these tissues. So we put them in the bath for about 3 hours, and then we started following them and measured the different contractility peaks that they have, or the peaks really. And we evaluated how much they contract. And in fact, of course, the controls seem to be contracting continuously. As you see, the frequency of contractions is about 4 per minute, peaks per minute. So it's kind of constant, while in the case of cryopreserved tissues, the 60, which is the green one, doesn't seem to be contracting that much. And the other thing that we observe is that it seems that although the contractility start increasing in the cryopreserved tissues, they seem to be kind of a spasm throughout so there doesn't seem, as you see in these two images on the left, on the right, that there seems to be some kind of peaks, but it's more like a spasm rather than an actual contraction bout. And then we did some histology to check what was going on with the tissue itself, with the muscle cells, what is happening. And what we observe is that there is a decrease in the percentage, or in the expression of connexin, in the production of connexin in these tissues, so in the myometrium. And the connexin is this protein that forms part of the gap junctions, which keep the communication between the cells. So what we believe is happening is that with the cryopreservation, we are breaking those connections. And although the tissue seems to be still, the receptors be active, and the tissue is still contracting, they cannot communicate to produce those nice peaks of contraction, but they are kind of contracting on their own. That's why you have kind of a spasm rather than a proper peak of contractility peak. And the other thing interesting in this one is, as you see, although we didn't measure it in this particular experiment, there seems to be a differential expression of connexin in the endometrium in controls compared to cryopreserved, which seems to also be reduced. So that may explain the other things about prostaglandin F2 alpha. So just to conclude from this series of experiments, and I said these are just the preliminaries, and we are hoping that we can continue trying to adjust some of these. We are working with people in Seville, which they think might be a good way of preserving muscle that might be the best way. So we saw that usually perfusion time between 45 and 60 minutes give us the best result. If you keep it longer, it causes more problems than it should. The endometrium, although the endometrium is still active, it seems that because of the way that we perfuse them, by the time the cryoprotectant reaches the endometrium might be already undergoing some damage or some degeneration. So one way that we think might be possible to do this is trying to perfuse the tissue using the vascular system, of course, the blood vessels, but also perfuse them through the cervix. So in that way, we might be able to recover also or we have two different ways of going from the lumen of the uterus into the wall of the uterus, and then from the vascular tissue as well into the wall, into the endometrium. So we also observe that there is a vascular damage, which there is an option that this is a mechanical problem, because when we observe the perfusion for 10 minutes without the cryoprotective solution, it seems that there is also this effect on kind of sweeping the endothelial cells. So it seems like they just drag the endothelial cells out, and that exposes the fenestra into the endothelial spaces, and that's where the 0.1 micron microspheres may be going. The other problem that we think it might be happening is that because we are perfusing through using the vascular system, the perfusion might be filling the vessels, the blood vessels. They might be filled with cryoprotectant solution at the time that they are frozen. So they are frozen in an open or in a distended space. And then once that distension disappears after thawing, that distension may also be not only affecting the blood vessels, but also affecting the myometrium that is surrounding those blood vessels. So that's why we see this change in contractility in the cryopreserved and controlled tissues. So one of the things that we might be looking into is trying to drain those vessels and see if that improves a bit the recovery of the myometrium, but we are not sure. Might be that is the case. And of course, as usual, all what we have been doing is in vitro. So we have done this several times, and it seems to be kind of a continuous response. So we need to adjust some of the protocols. But exactly the same thing happened with the ovary when we were doing this. There seems to be that you never reach the perfect state, but once you attempt a transplant, usually the cryopreservation seems to work. So that's what happened with the ovary. And I think until we cannot run, until we run the in vivo cryopreservation and transplant and see if that works, then probably a lot of the in vitro results that we have may be quite good at explaining what may happen later on. But until we do that, we won't have our answer if this would be working or not. So I just would like to thank all the people that has been involved in this project, specifically Professor Bruce Campbell, who is retired now, but he started this program of cryopreservation in Nottingham, and then the students and the technicians that have been involved in this, and to Dr. Matthew Elms, which provided all the contractility work. And thank you very much for the invitation, and any questions, I'm happy to answer. Thank you.
Thank you very much. That was a fascinating presentation. There are actually no questions in the Q&A, but I would have a question if I'm allowed, and that would be your connexins. Do you think that they would recover once you have transplanted actually the organ?
I don't know really. I think until now, because of the damage that we are seeing, it might be quite difficult that they will recover. And what may happen is that you end up with a tissue that might survive. So the perfusion might come back, but then the tissue might survive, but probably will never recover the actual function using our current protocol. So I think if we fix this a bit and we reduce the damage into the muscle, then there is a chance that this tissue will recover. I know freezing muscle is one of the biggest challenges at the moment. I've been talking to Dr. Risco in Seville, and they've been trying to freeze hearts, and it's the same. It's trying to freeze muscle, and muscle that contracts and that interacts, that coordinates is the problem. And I think if we manage to reduce the damage to the muscle, I think there is a scope for the connexins to recover, the gap junctions to recover, given enough time, and some of the contractility might come back. Not sure if full contractility, but at least some might come back. That's my thought.
Great. Well, we have already we're already running over time, so I don't know if we have time for more questions. Please send me some in the chat. Otherwise, I think we are done with this session.